Well, hello, and welcome to Bio 211. This is Anatomy and Physiology 1. It's a section at Norwalk Community College, and we are starting and completing today Chapter 12. And what we're doing is we're looking at an overview of the nervous system itself. And you can see here that I find the chapter to be very exciting. And yes, it's an intentional pun. Don't roll your eyes, because what you're actually doing inside this chapter is not only are you thinking about the three parts to pay attention to, which is one, can you build a neuron, right? Can you actually do that? By the time you finish this chapter, you should be able to build. The textbook focuses on a multipolar neuron. You'll see what I mean by that in a minute. That's not what I wanted to do there. Can you build a neuron? What makes it function electrically? So by the time we finish, what is it that's unique about what you built that will allow it to function electrically? And electrically, of course, means which ions are moving and what is the consequence of those ions moving. And believe it or not, your nervous system signals between neurons, in some instances electrically. If we think about back to chapter 4, we can create gap junctions where you have continuous electrical signaling between cells because of the connection proteins, right? But what we're going to do here is we're going to regulate the release of neurotransmitters to engineer a delay between electrical signaling in cells. Okay, so you're going to see when we start drawing this what Mother Nature's doing in her really ingenious and exciting way. She's neat. She's figured out, hey, these electrons, the ions, the charges that are moving across the surface of cells can move too fast. And I need to be able to tell them exactly where to go and what to do. And to do that, I'm going to use things that we're going to call neurotransmitters. Okay? And to bring that back to chapter 3, neurotransmitters are simply ligands to receptors that you find on cell surfaces. Okay. Of course, there are going to be different ones. Some will be excitatory, some will be inhibitory. And that's the work of this chapter. But by the time you finished it, man, you've done a significant amount of work to understand how it is Mother Nature can make a responsive organism that can survive depending upon the environment it finds itself in. So, along with the endocrine system, we actually use the nervous system to control the conditions within the limits that maintain health and homeostasis. Okay? When we're looking at inside your textbook, or in this case, this video presentation thing, you can see here in this cartoon of a semi-correct organism in anatomical position. You can see the central nervous system is the brain and the spinal cord. The peripheral nervous system makes up the cranial nerves, the spinal nerves. Those ganglia, which are the cell bodies of neurons outside of the central nervous system, and enteric plexus and sensory receptors. Excuse me. So Mother Nature has a mechanism to collect information from the outside of the body, send it to the central nervous system, where the central nervous system will then process that information, and then send motor signals outward again. So you can see what we're doing is, even at the very beginning, is we're reinforcing this idea. Let's go for unique colors here. Let's go with blue first. Blue is going to be the sensory input that travels from the peripheral nervous system. Okay, it's going to activate. Let's go for purple. Where are you? Fantastic. Purple to some control center activity, which is going to make the decision of what's going to happen as a motor output. And then we're going to have some motor output, which is then going to cause activation of, whoops. Let's go like that. Wham. Activation of some effector. So I'll put an E down here, right? Cool. So that looks familiar, doesn't it? Think back to chapter 1 when we're talking about feedback systems. In fact, what I'm going to make sure you walk away understanding today, or sitting day today understanding, is that feedback systems inside the nervous system are positive feedback systems. Okay, so what are the major structures? Let's get rid of the cartoon. The major structures are, there are 12 pairs of cranial nerves, okay? There are 31 pairs of spinal nerves. Those ganglia are located outside the brain and spinal cord, and they are small masses of nervous tissue. But what you need to remember, of course, is they are the cell bodies of neurons. Okay, so let's make sure we do that. Okay, and what we're going to do with this information right here, right? I think you guys remember I drew something, or I have drawn something that looks like this. The central nervous system is a ball on top of a stick, right? And if we have 12 pair of cranial nerves, look, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 on one side, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 12 on the other, and now 1, 2, 3, 4, 5, 6, 7, 8, 9. You get the idea. 12 pair, 31 pair, and this is how you start building the intricacy of the nervous system itself. Okay, so you can see it's a basic wiring diagram. And even though this cartoon is fundamentally crude, that's what I want you to understand. Mother Nature needed to figure out a way to develop something that was much less crude. Okay, let's get rid of that cartoon. Let's move on. So how does it function? What is it actually doing? Part of its responsibility is to collect information that we call sensory information. And we have big fancy words for that to describe that. And you want to start learning those fancy words, okay? Because afferent neurons collect sensory information. Motor neurons are efferent neurons, and they send motor signals, okay? Somewhere in between, we are going to use interneurons, okay, which were the purple ones a moment ago, to create some form of integration. Inside, probably a little way too early here, and they put association inside your textbook, right? You're going to see these are specific types of neurons inside chapter 14. But for right now, again, if I was to draw this for you, I'm just going to use one color. I'm just going to use black here because I don't want to waste time drawing between all these things. Well, there's your sensory input. There's your interneuron. And there's your motor output. Remember, that would be blue. That would be purple. And that would be red. For individuals who are colorblind, essentially all sensory neurons are afferent neurons. All interneurons, okay, or association neurons are the integration site. Okay, and then you have the motor output. Sorry if it seems like I'm belaboring this, but this is literally the physical structure and the nomenclature that we use, or the naming system that we use, and everybody's expected to understand it, so you are as well. Okay. So let's go like this. And a moment ago, I said, look, we have a central nervous system and a peripheral nervous system, and I drew those things for you. How did I do it? I drew a ball on a stick, and then I put 12 pair of cranial nerves, and I put 31 pair of spinal nerves. Okay? How does it work? That's the hard part, right? This is a cartoon of a multipolar neuron, and I believe I've probably had you guys draw this already. Okay? And what you're going to see is there is a repeating pattern on all multipolar neurons. I will sort of block this off for you so you can see that, look, here is one functional area, which is the dendrite in the cell body. And then over here we have another functional area, which is the axon and the axon terminal. Make sure I do this properly. And then over here we have this thing that's called the axon hillock. So you'll notice I have 3 different boxes. And that's the way I want you to approach learning this. By now, you've hopefully thought about it enough to know that there's going to be a unique set of responsibilities at the dendrites in the cell body, a unique set of responsibilities at the axon hillock, and then a unique set of responsibilities at the axon itself. There are some differences that I want you to pay attention to. So you'll notice inside the cell itself, instead of having free-floating ribosomes and rough endoplasmic reticulum, you actually have structures that are called Nissl bodies, which is where protein transcription and translation will occur. Okay? And you'll also have, inside of it, you're going to see these filaments. You actually have long tracts of structures that are going to allow for the deformation of the cell to maintain this really elaborate structure. Okay? And this is just the tip of the iceberg in terms of what neuron shapes can be formed. But now you know that there are three basic parts to it. A part where information can be collected, information can be interpreted, and then you can have some effector output. Do you see what I just did there? So if the entire nervous system is built on sensory input, integration, and motor output, each individual neuron has sensory input, I'll put an S there, integration, and effector or motor output. Okay? Yes, no, maybe? Okay, cool. So how does it all work? Let's get rid of the cartoon. Yay. When we're looking at the actual neurons themselves, you want to understand that they are excitable. And excitable means that they actually have the ability to produce potentials. Let's just get rid of action potential because that's confusing. And potential just means that they can either increase or decrease charge at the membrane. Okay? And that's what we mean by excitability. Right? The cell body itself usually has a single nucleus. In your practical, you saw there was probably a nucleolus in there. Nissl bodies, this is the rough ER as well as the free ribosomes I just mentioned a moment ago. Okay? Neurofilaments give the unique and elegant shapes that you saw. Microtubules, filaments right here, right? Okay? Microtubules, okay, are going to allow us to move materials. We're going to come to that in a moment when we start talking about how it is we use fast and slow axonal transport. Okay? And processes are those structures that are presented off of the actual cell body itself. So in this case, both dendrites and axons on the axon terminals would be processes. Okay? What are the different types of structures you can see? We're going to spend most of our time thinking about multipolar neurons and unipolar neurons in chapters 12 and 13. In chapter 17, we spend a fair amount of time thinking about bipolar neurons. And this is where I'm going to point out that chapter 17 will be on the final exam, but I will teach it so that we use it as a review tool. Okay. So how do things move inside of the cell? We have this thing that's called axonal transport, which means materials can be made almost anywhere inside the cell, be put onto the microtubules, and they can move throughout the entirety of the cell. There are two types. There's slow axonal, okay, which is in one direction. Okay. And you have fast axonal transport. Okay. And these can move in both directions. That's why they say either direction. Okay. Underneath here. And it's for the movement of materials. And I tend to think about it use or for use of NT movement. And for recycling inside the cell body. Okay? And NT, of course, means neurotransmitters. So we're going to make and break neurotransmitters apart. Cool. So, once you understand that, what I want to do is I want to introduce this idea that Mother Nature decorates your body with sensory receptors, right? So, Control B. Oops, that's supposed to be the whole thing. Well, whatever. Makes no difference. Sensory receptors that are the dendrites of unipolar neurons. Okay, so what does this mean? Here's our color scheme, like for the, I don't know, second or third time. Sensory input. Remember, sensory input inside of our model is blue. And it's going to travel in to then activate an interneuron so far, right? Purple, where processing is going to occur, or interpretation will occur, or whatever you want to call it. And then we're going to use red for motor output, right? I need you, and you need you, more than anything else, to understand that, look over here, that Mesner corpuscle, that Merkel disc and Merkel cell, the Pacinian corpuscle, and those free nerve endings. And notice right now we're giving you a specific name. These are called nociceptors for pain. Okay, these are all the dendrites of unipolar neurons. Okay, which means, take a look here. Here's the cell body. And in the human body, the cell bodies of each of these unipolar neurons are located adjacent to the central nervous system inside the dorsal root ganglia. And you'll see why that is important later. Okay, so that means Mother Nature collects all sensory receptors from unipolar neurons into unique locations, and you can begin to see how Mother Nature has said, okay, I'm going to use this repeating pattern to collect different types of information. Pressure, skin pressure, deep pressure inside tissue, pain, free nerve endings. And regardless of the type, it still travels inward, it's still going to be interpreted, and there's going to be some motor output, should motor output be required. Now, your textbook sort of switches gears here, right? So if these are neurons and some of their basic structures, and the fact that there's a repeating pattern, you have sensory input, interpretation, and motor output, she meaning Mother Nature, couldn't function unless she had supporting cells, okay? A support staff, and those support staff structures are called glial cells. And inside your textbook, they introduce the concept of glial cells by building a interface or an interface between blood and brain. So what you're looking at here is the actual structure where we have a ventricle here. The ventricle is the inside part of the brain. Meninges, pia mater, which cover the brain. And then in between these two pieces, we're going to have neurons, vasculature, and a variety of cells that are supporting the activity of the central nervous system itself. Okay? In this cartoon, you have the most common types of glial cells, which are astrocytes. You're going to see that there are different types. There's a fibrous astrocyte. There's a protoplasmic astrocyte. Okay? You're also going to see these microglial cells. Inside your textbook, they're green. Inside this picture, they're red. You're also going to see that you actually have structures that are surrounding the axons of neurons. And because we're inside the central nervous system, you're going to notice they have a unique name. And those are oligodendrocytes. If they're inside the peripheral nervous system, they're named Schwann cells. Okay, so what are we doing here? Whoa. What happened there? Getting all excited about teaching this. Okay, cool. So what's actually going on here? So what Mother Nature's doing, she's allowing diffusion to move into this space, and plasma is going to be pushed through those walls, which then becomes interstitial fluid. So that interstitial fluid is now going to be filtered through the last of the glial type cells. Even though it's epithelial-like, it takes materials up into that ependymal cell and then pushes them through to the ventricle side here, where I want to make sure you understand the central nervous system has its own circulatory system, and the fluid that it pushes through it is cerebral spinal fluid. So what you're looking at here is your textbook is trying to be, I think, a little cute in saying, oh, look, if we look at how we use all of these glial cells, you can get the general idea that we can have a separation between the outside part of the body and the inside part of the body. Well, yeah, every part of the body is broken up like that, though, so it makes no difference. This is actually really complex. And in some ways, they're oversimplifying it. But what we can do is we can turn it into a teachable moment. You want to know what protoplasmic astrocytes do. They break down materials. They metabolize spirulous stuff that doesn't belong inside the space here. Okay? Microglial cells do exactly the same thing, meaning they literally break down the cells that should be destroyed. Okay? Those ependymal cells are filtering the interstitial fluid that traveled through here to create CSF. So let's put this here. CSF. And that CSF is now on this side of the ependymal cells. Okay? And that's a pretty good place to stop. So we've now introduced what is an excitable cell, what are neuroglia, and now we can start to assemble our nervous system.